Good morning for those who are in the Americas, and good afternoon for those who are in Europe, and good evening for those from South and Southeast Asia. My name is Umaharan. I am the director of the Cocoa Research Center of the University of the West Indies. Both on behalf of myself and the entire staff and students of the Cocoa Research Center, I'm extremely pleased to welcome you all to the second of a two-part webinar series. Last week, we had our first webinar entitled Improving Cocoa Profitability with emphasis on quality and food safety. Many of you all would know that the Cocoa Research Center is part of the University of the West Indies that has a mandate to support 16 countries in the Caribbean. So although we are located in the Twin Island state of Trinidad and Tobago, we have programs in Belize, Jamaica, Haiti, Dominica, St. Lucia, and Trinidad and Tobago. The Latin America and the Caribbean region is the largest producer of fine or flavored cocoa, and hence we are interested in improving the quality, flavor diversity, consistency, and food safety, and supporting the entire Latin America and the Caribbean region through outreach. Hence, in the first webinar a week ago, we dealt with quality and food safety aspects, which are critical to this particular region. In addition to the regional mandate, as the custodians of the International Cocoa Gene Bank, the Cocoa Research Center also has an international mandate. Hence, normally when I introduce the Cocoa Research Center, I introduce it as a regional institution situated in the Caribbean, but with a regional and international mandate. Today's presentations are largely about this international mandate. How do we leverage the genetic resources that we have in the International Cocoa Gene Bank to support the profitability of the global cocoa industry, be it in West Africa, Southeast Asia, the Americas, or in the Pacific? In the second webinar, we want to talk about the global mandate in detail. And to deliver on this mandate, we need to develop collaborative measures. So we'll also hear about some of our collaborations with other institutions during our presentations. As I had done in the last webinar, I would also like to give a presentation at the start to outline the strategy of the Cocoa Research Center so that all the other presentations that follow will have a perspective. So I'll go on to my presentation. I'm going to open up my presentation now. Give me a second. Okay, so today's presentation my presentation is going to outline the strategy that we are building at the Cocoa Research Center with regards to developing a pipeline from the gene bank to the cocoa farms around the world. Right, so that's the topic I'm going to talk about. So I go to my first presentation, the first slide. You will know that the Cocoa Research Center is the custodian of one of the international cocoa gene banks called the International Cocoa Gene Bank Trinidad. And there's a second gene bank also in Costa Rica at the Catier, which is also another public gene bank. The gene bank in Trinidad and Tobago is about 100 acres in size and has 2,300 accessions that have been collected from different parts of the Latin America and the Caribbean region. Now, this region is regarded as the center of diversity for cocoa. Now, each of those 2,300 accessions are clonally multiplied as rooted cuttings and are planted in plots of 4, 8, or 16 trees, depending on the accession. These accessions are largely heterozygous in nature because of their self-incompatibility system. Now, the genetic resources in the gene bank have to support countries around the world farmers around the world. So I've listed some of the requirements of cocoa farms around the world. And these include, first, they would like to have varieties that are highly productive so that it contributes towards a profitable cocoa industry in their countries. And we need to really improve the tree architecture, self-compatibility system, and usually we need to have heterozygous but homogeneous population. So this is something for the farms. In addition to having these varieties, we need to also ensure that there is some level of diversity among the varieties distributed to ensure that there is some level of resilience and we don't depend on one variety throughout the country. The third aspect that we need to look at is local adaptation. Local adaptation to the agroecologies that are peculiar to each of the countries. And the agroecologies are characterized by the specific biotic stresses that they experience, such as diseases and pests, the kind of abiotic stresses that they experience, example, drought, the cropping system that is peculiar to the country. Some countries have more orchard type systems, which are monocultural. Some countries have more agroforestry type systems, and others have a multiple cropping system. And then the management of the farm, whether its level of management is very high or low, and I'll talk about this in a minute. And then finally, the kind of market that you are producing the cocoa for, whether it's the fine or flavor cocoa market or it's the bulk cocoa market. And depending on that, the flavor and the butterfat profiles have become very important. So on one hand, you have the genetic resources in a gene bank. And on the other hand, you have these cocoa farms throughout the world in different countries that have different peculiar requirements. And so what we need to therefore do is that try to fill the gap between the two by building what we call a pipeline. And that is what we are going to talk about today. How do you build a pipeline for genetic resources to move from the gene bank to the cocoa farms around the world? And to do this, we have to extend our services in the gene bank beyond what we do now to create part of the pipeline. And the cocoa industries around the world, the countries that is, have to also extend some of the things that they do to also fulfill part of the pipeline so that we complete the pipeline from gene bank to the farm. So traditionally, the gene banks are set up for conservation, to conserve the genetic resources. And usually they have the following functions. They would acquire germplasm from the centers of diversity so that they have a collection that is well represented by the diversity that exists around the world. Secondly, they would characterize these collections, and usually in the Cocoa Research Center, we characterize it based on morphological characters. We use about 22 heritable characters to characterize them. And then we also do a molecular characterization using a number of SNP markers to identify the gaps in the collection so that we can continue to do collections to improve the diversity and the representation of the gene bank. Most importantly, we also do evaluation. Now, you cannot have a collection that is not properly evaluated because evaluation provides the opportunities for improvement and utilization of the genetic resources. So here are some of the traits for which we are evaluating: yield components, disease resistance, cadmium uptake, tolerance to high light intensity, drought tolerance, flowering time, pod development period, quality, and so on. So these are some of the things that we look at. And then all of this information that we have collected has to go into a gene bank management system and shared with the international, globally. And this is done through the International Cocoa Gene Bank Germplasm Database that is held at the University of Reading. And then finally, we have to duplicate the collection to ensure that the collection is safeguarded. Now, this is what we regard as the traditional functions. Now, if you want to build a pipeline, what else do you have to do? So one of the things that we have started doing is to establish a marker discovery platform. And to do this, we have collaborated with a number of our partner institutions, and we have set up populations for genome-wide association studies. So we have set up a population of 500 accessions that have been genotyped or sequenced, and we have developed a pipeline for GWAS. So all the characteristics that we evaluate on these accessions can be passed through this pipeline to develop potential markers. And then we have established a number of biparental progenies to validate some of the markers coming out of the GWAS platform. And these will constitute a marker discovery platform. But following the marker discovery, we need to now establish what we call a genomic selection platform based on these markers, validated markers. And we have established a number of biparental progenies that we would like to screen with this marker panel so that we can create what we call enhanced populations. Now, the enhanced populations, at the moment, we have two populations. One enhanced for black pod resistance, and another one that has been enhanced for pitcher food resistance. But you could think about different enhanced populations for different parts of the world, depending on their peculiar requirements. And these enhanced populations have certain characters fixed to that population, but they also have inbuilt variability in these populations so that they can be adapted in the end user countries. So this is what we would like to do in building the pipeline. So this is part of the pipeline that the gene banks would have to do. The other part of the pipeline is what I think the countries have to build. And that is to correctly characterize their receiving environment. Because if you characterize your receiving environment accurately, then you could receive the correct enhanced population for introducing into your country. So usually the enhanced populations are transferred through an intermediary quarantine that is established at the University of Reading. And from there, it would be transferred to the end countries, where local adaptation trials can be done in different agroecologies to select the best material that can then go to the farms. So the farmers therefore receive material that is more adapted to their conditions, and therefore it results in increased productivity for the farm. It lowers the risk associated with producing cocoa, and therefore it results in lower cost of production. So the increased yield coupled with lower cost of production will result in increased profitability. So this is the role that the pipeline will have to play in terms of developing still systems. So what I've also said is that traditionally, in most of the cocoa growing countries, including in Trinidad and Tobago, we develop varieties and we introduce the varieties to farms regardless of their local conditions. And should we be doing that? Should we have a more nuanced deployment of cocoa genetic resources rather than having this sort of blanket way of applying varieties? And this would contribute towards more precision agriculture. We call it precision agriculture because you try to move varieties to the agroecologies depending on their needs. So this requires that countries should map their agroecologies. Within each agroecology, they should identify cocoa growing communities. And for each of the growing communities, they need to identify the risk factors associated with it, the constraints, and the cost drivers. And therefore, once you understand this, then you can introduce the right kind of variety to mitigate some of these risks and reduce the cost of production. Then you need to map the farm or farmer requirements. What kind of cropping system are they practicing? Is it an orchard type monocultural system? Or is it a multiple cropping system? Or is it under an agroforestry system? And so on. And the kinds of varieties we introduce have to be sort of nuanced to meet the needs of these systems. The level of management of the farmer is also very important. You cannot really provide very rigorous material to a farmer who has a very low level of management, because it's going to only contribute to further reducing the yields that they obtain. So you need to really depend on the level of management, be able to support the farmer with the right kind of planting materials so that you suit the farmer needs. And finally, the market. You need to really look at what is the market that the farmer or farm is supplying, and what sort of marketing system that they are using, so that you can have a better nuanced way of supporting them with the kind of varieties that they need. And so as you go from this blanket thing to more nuanced application, the profitability of the farmer will increase. And this will reduce what we call the yield gap. And in the world, if you look at cocoa, you will have materials that can produce 2 tons to 3 tons per hectare. That's the potential yield that they can get, but they get maybe 200 to 300 kilograms per hectare. So that the yield gap is tremendous. And one way of reducing it through this nuanced way of applying it. So in Trinidad and Tobago, we have started this exercise. We have mapped 6 agroecological regions in Trinidad, which are shown here in the different colors. And each of those agroecological regions, we have further subdivided them to cocoa growing communities. And we are assessing the risk profile and the constraints within each of these agroecologies so that we can supply them with the appropriate material or genetic material to be grown in those areas. So this is something in progress. We are not going to be presenting on this today because we haven't completed the exercise, but we hope in the future we are going to show you some of the findings from this work that we are doing. So that takes us to my conclusion. We consider the International Cocoa Gene Bank as a wishing well. Make a wish, and you throw in some effort behind it, and you could get solutions to every problem in the cocoa industry. So this is a tremendous resource that we have that has to be utilized. And for utilization of this resource, we need to build a pipeline from the well to the cocoa fields of the world. And that is the pipeline that we were talking about today. And the Cocoa Research Center's efforts are towards modernizing the International Cocoa Gene Bank, towards building this pipeline so that the benefits of the genetic resources could be had in the end user countries. And to do this, we cannot do this alone. We need our collaborators to work with us on it, both the funding agencies as well as the collaborating research institutions. I'd like to end this with a quote from Susan McCooch from the Cornell University. You know that she's a very eminent researcher who has worked a lot on rice. And this is what she has to say. Gene banks need to take on a new life. Previously seen as warehouses where seeds were diligently maintained, but evolutionarily frozen in time, gene banks should transform into vibrant research centers that actively investigate the genetic potential of the holdings to the benefit of the society. In today's presentation later today, you will hear presentations about the renovation of the Cocoa Gene Bank, the work that we are doing to improve the standard of the gene bank. We'll talk a little bit about the work that we are doing on characterizing the gene bank. We'll talk about the evaluation of the gene bank for various characteristics. Today, we could only concentrate on 2. The other future presentations, we'll talk about the others that we are working on. Fidelity and the duplication of the gene bank. We'll talk about the DNA fingerprinting exercise and information that is bringing to the fore. We're also talking about the mapping of diversity that we have done in Trinidad and Tobago. The other items we will reserve for another day, but I would like to thank you all for listening to my presentation. I want you all to give support to some of our researchers and students who will be presenting today. We'll have presentation from 4 different groups today from genetic resources. We have 2 presentations one by Antoinette Sanka and Reena Kalu. From the pathology, we have from Romina Umaharan. And the physiology, biochemistry, you have Adrian Lennon, who will be presenting. And from the genomics section, we have two presentations from Lambulit Motilal and Trishana Maharaj. So I'd like to take this opportunity to thank all the funding agencies, particularly the government of Trinidad and Tobago, who have supported us over the years, and the Cocoa Research Association of the UK, which has supported the Gene Bank and the work that we are going to be presenting today. The Cocoa Research Association of the UK is supported by funding from Mondelez, Mars Wrigley, Barry Callebaut, Olam, Cargill, Etad Chocolates, and Vitecas. So we thank them all. And also I have to recognize our collaborators from the University of Nottingham, University of Reading, both from UK, SRAD from France, who have had a very long journey with us, and USDA ARS, who has also been working with us on genetic fingerprinting. Thank you all.